Hello, I'm Dr. Salim. Today we'll be doing bilirubin metabolism, hyperbilirubinemia, unconjugated conjugated bilirubin, cellular injury marker. So, bilirubin is produced when red blood cells expire and they have to be catabolized and excreted from our body. Red blood cells are produced from hemopoietic stem cells in the bone marrow. Alright, so after 120 days, RBCs expire, right? And new red blood cells have to be produced. Red blood cells, they have high regenerative capacity. They reproduce frequently. So examples of they are called labile cells. So another example we can get is epithelial cells. They have high regenerative capacity. We have other cells that doesn't have high regenerative. We have labile cells which have high regenerative capacity. Then we have stable cells. Stable cells regenerate when they are stimulated. Example is hepatocyte. Then we have permanent cells. Permanent cells have very low regenerative capacity. Examples of permanent cells are neurons. So after 120 days, RBC will be engulfed by the splenic phagocyte. So it will be engulfed by splenic phagocyte and it will be broken down into hemoglobin. Hemoglobin will further be broken down into heme and globulin. Hemoglobin is a protein, so it will re-enter circulation. Heme is made up of iron and unconjugated bilirubin. Iron will then be re-enter circulation to be used for the production of RBCs. Unconjugated bilirubin here will bind to albumin. Unconjugated bilirubin is lipid soluble. So what you have to know is that hemoglobin will be broken down into heme and globulin. Globulin is a protein and it will enter circulation. Heme is made up of iron and unconjugated bilirubin. Iron will re-enter circulation. It will be used for the production of new RBCs. Unconjugated bilirubin is lipid soluble. So it will be the reason why I'm stressing on this is that hyperbilirubinemia in neonates greater than 20 mg per deciliter can cross the blood and brain barrier and cause kernicterus. Blood and brain barrier permits only molecules that are lipid soluble. So drugs that are used in CNS, for example levodopa, they are all lipid soluble. Only lipid soluble drugs or lipid soluble molecules are able to cross the blood and brain barrier. So since unconjugated bilirubin is lipid soluble, high levels of it can cross the blood and brain barrier to cause kernicterus, seen in children with hyperbilirubinemia or jaundice. So since it's lipid soluble, it will be carried to the liver by albumin. We say that many molecules bind to proteins, example albumin. So it will bind to albumin, it will be carried to the liver. In the liver, now, unconjugated bilirubin will be converted to conjugated bilirubin by glucuronide transferases. It's an enzyme, glucuronide transferases. So it will be converted to conjugated bilirubin. Now, conjugated bilirubin is water soluble. Unconjugated bilirubin was lipid soluble. Conjugated bilirubin is water soluble. So now it's water soluble, it can be excreted freely together with other bile salts. So conjugated bilirubin will travel through the common bile duct into the intestinal wall, into the intestine. In the small intestine, conjugated bilirubin will be converted to urobilinogen by intestinal bacteria. Right? It will be converted to urobilinogen by intestinal bacteria. 10% of urobilinogen will re-enter circulation to the kidneys and it will be excreted renally. 90% of urobilinogen will be oxidized. It will be oxidized into sterocobalamin, and sterocobalamin is what gives yellow pigmentation of feces. So you have to know that this is a very simple pathway. So from heme will be broken down into unconjugated bilirubin and iron. Unconjugated bilirubin to the liver will be carried by albumin. From the liver, unconjugated bilirubin will be converted to conjugated bilirubin by glucuronide transferase. Now it's water soluble, so it can be excreted with other bowel component like bowel salts. So it will be transferred, because water soluble, into the small intestine. Conjugated bilirubin will be converted to urobinologin by intestinal bacteria. 90% of 10% of urobinologin will re-enter circulation and go to the kidneys and will be excreted renally. 90% will be oxidized into sterocobulin, and sterocobulin is what gives our feces the yellow pigmentation. So one thing you have to know, I've simplified it here. Erythrocytes, in medicine it is important to understand. That's why I took my time to explain this to you before I go to the points that I've made. So let's go to the points now. Erythrocytes are produced in the bone marrow through erythropoiesis. Alright. Immature RBCs, also known as reticulocyte, and then mature RBCs, erythrocyte. RBCs carry oxygen. Here it is important that red blood cell, the main function of red blood cell is to carry oxygen. That is why hypoxia, the most common cause of hypoxia is ischemia. Alright. The most common cause of hypoxia is ischemia. It is very, very important. The most common cause of low oxygen levels in the blood in our body is reduced blood flow to a tissue. So it's important. So RBCs carry oxygen. So the life cycle of RBC is 120 days. Phagocytosis takes place in the spleen and will be degraded into hemoglobin molecules. Heme and globulin, as I said, heme is made up of unconjugated bilirubin and iron. Globulin is a protein. So heme is made up of unconjugated bilirubin and iron. Unconjugated bilirubin will be carried by albumin because it's lipid soluble to the liver. It's lipid soluble. Alright. Unconjugated bilirubin will be converted by glucuronide transferase. So I made it here. Conjugated bilirubin here, this one should be water soluble. This is an error. This one should be water soluble, and this one, unconjugated bilirubin, should be lipid soluble, and conjugated bilirubin should be water soluble. So after conjugation, the end product that you get as conjugated bilirubin is water soluble, so it can be excreted, it can be moved, and it can spread through the intestinal walls. So conjugated bilirubin is transported via the bowel to the small intestine. Alright. Conjugated bilirubin will be converted to urobilinogen by intestinal bacteria. 10% re-enter circulation to the kidneys and it will be excreted renally. 90% will be re-oxidized into sterocobulin, which gives feces its brownish pigmentation. Alright. This is a very simple pathway. So the pathologies that we can make from here, I told you that in neonates, pediatrics, hyperbilirubinemia in neonates. So sometimes physiologic jaundice occurs maybe after 3 to 5 days. It's normal because this enzyme, the liver is not matured enough. So there is a deficiency of this enzyme, glucuronide transferase. So in neonates, sometimes there will be jaundice in the first few days of life, which is normal because the liver is not matured enough, all right, to convert unconjugated bilirubin to conjugated bilirubin, which is normal in life, all right. But jaundice, which the bilirubin levels is greater than 20 milligram per deciliter, can cause kernicterus. Alright, can cross the blood and brain barrier and cause kernicterus, which is very fatal. So that's what I want to know. I want you to know. And then someone also asked me that the importance of inflammatory markers, for example, ALTs and ALTs. So I just want to and talk about that small. So when we are doing cellular injury, all right, in cellular injury we have reversible and irreversible injury. All right, so in irreversible injury, I'll talk about reversible injury in another video. So in irreversible injury, when there is an irreversible injury, lack of ATP will lead to severe membrane damage, all right, which will cause influx of calcium into the cells. Calcium acts as a second messenger and activates enzymes and protein, which is seen in injury. So calcium, it acts as a second messenger, so it activates enzymes and protein which are seen in injury. So when there is an inflammation, for example, when there is an injury, myocardial ischemia, when there is a bile duct injury, when there is a liver injury, you see that inflammatory markers and enzymes and proteins are elevated. It's due to this mechanism. All right, so one, myocardial infarction, you see CKMB, all right, creatinine kinase MB, troponins I, C, and T. Then we go to hepatocellular injury, for example, acute liver failure. They will be raised in AST and ALT. So AST and ALT, they are enzymes that are in the liver, aspartate transaminase and alanine transaminase. Pancreatic duct injuries, for example, gallstones, there will be increase in alkaline phosphatase. Pancreatic injury, there will be increase in pancreatic amylase and pancreatic lipase. Kidney injuries, raising creatinine level. So anytime there is an injury to any of these organs, that's the reason why when there is an injury to any of these organs, we look at these markers and these enzymes. When they are raised, it signifies. So that's the reason the person specifically asked me about AST and ALT. So when there is a hepatocellular injury, maybe acute liver failure due to maybe paracetamol toxicity or mushroom toxicity, all right, there will be increase in aspartate transaminase and alanine transaminase, because in irreversible injury, reduced APT will cause influx of calcium inside the cell, and calcium acts as a second messenger and it will activate these enzymes and proteins. Right, this is a short video. I hope you enjoyed this, and thank you very much.